Testing, 1, 2. Good evening, ladies and gentlemen. Welcome back to Reef Lounge. Look at our beautiful microphones. What are you laughing at? You taking the piss?
Nope.
Well, what am I meant to do when you're sitting there on your phone?
I just had my phone in my hand. I'm sorry.
He's always got his phone in his hand.
Yep.
It's becoming a running joke,
It is.
isn't it?
It is.
2 running jokes. Davey's always on his phone, and Davey loves goldfish. You do. He admitted it the other day, folks. I don't know if you saw that on Facebook.
I never said I liked goldfish. I said I hate goldfish.
Can I? It's something really, really funny.
Did you know Adam swings the other way?
That's not what I said. That's not what I said. No, it isn't.
It is.
No,
it is.
It's not what I've got to stop messing. I'm conscious of the fact that I'm developing a habit of moving this microphone around all the time. Need to stop doing that.
I hear boom.
It's a hard life.
It is a hard life. So, Adam, why are we here?
Why are we here? Well,
because,
we're recording a podcast.
Yeah, and we kind of messed up a little bit, didn't we?
What kind of?
Yes.
We are learning our trade when it comes to putting together audio-visual entertainment for the good people out there.
Yep.
And we had some guests on.
We did.
A couple of 3 weeks ago now, if we're honest about it.
It was the last bank holiday.
No way. Was it a month ago?
So it was a month ago.
Who did we have on, Davey?
So we had the beautiful couple from Scireef. We had Samuel and Marine. We did.
However, we made some itsy-bitsy mistakes with the audio recording, and it wasn't perfect.
Yes, it wasn't.
And since then, I have gone on a deep rabbit hole style learning journey about how to use AI engines to edit audio and make the audio something that was listenable to. Because, to be honest, it was a fantastic podcast. There was some wonderful content there.
There is.
They're great guys to talk to. We're going to do plenty of work with them in the future.
Yep.
And rather than not put this out, we have got it as good as we can get it, and we're going to put it out.
Yeah. So this is literally just a little intro, just to let you know the sound mightn't be that great, might be a bit choppy in points, but
A little bit.
it's okay.
There were issues a little bit that weren't in our control, like the internet connection was not as stable as it could be.
Yeah.
Nothing we can do about that. That's just part and parcel of talking to people and doing phone or mobile type recordings of interviews and stuff like that. But look, it's good content, and I kind of get the feeling that this is going to be another one of those ones that we're more critical of than other people, and they're going to go, it was fine.
Yeah, no,
but realistically,
a lot of people do say,
I know.
Yeah, it is fine.
But there's a lot of podcasts out there these days. There's a lot of people doing them. They're very good quality. People are very good at it, and we want to put ourselves in that space.
Yeah.
So we are personally hypercritical about what we do a little bit.
Yeah, we're just trying to make it better.
Yeah, I think we are.
Because it might be fine now, but in a couple of weeks when you're watching it, you might look at it and go,
yeah,
oh man, it's still doing that.
And I know it's going to happen. We're going to look at one of these in 12 months' time. We're going to at some stage click back to the first few that we did and went, bloody hell, did we put that stuff out?
Yeah, we
will.
Awful. Yeah. What are they talking about?
Yeah, I know.
Pair of idiots.
Yeah, why is it all bobbly and why are they so quiet and why do anyway.
Yeah.
So anyway,
why did they say that goldfish are great in a reef tank?
In a reef tank? I don't know about a reef tank.
Yeah, well, what are you messing? Boys, that was a joke.
Don't keep goldfish in your reef tank. If you want a goldfish, give me a reef tank. Come buy an Antheus.
Yeah.
That's near enough a goldfish, isn't it?
A sea goldie.
Sea goldie.
Is a wreckfish.
Is it?
Which is a lardertail.
But does it look like a schmunkin? Stop looking at the schmunkins.
Kind of. No,
can't keep your eyes off those fish.
No, really.
Anyway, guys,
quite absolute.
Yeah, I know I am. I'm sorry. Quite enough from me and Davey. We're going to go over to the interview, and I hope you guys enjoy listening to Scireef. Fascinating guys. We're going to be doing work with them ongoing. Davey's talking about getting corals in from them. We're going to be having some of their stuff knocking around the shop hopefully pretty soon, aren't we?
Not going to be getting corals.
We are getting corals.
We might even have pictures out before people see this podcast.
Really? Wow.
Yes.
Okay, so it's on the way.
It's on the way.
And that's so exciting. I'm not going to. I want to talk about it because the way that the corals come about is so amazing. That anyway, listen to the interview.
It is great. It's a great chat. I have to say.
Watch the chat. Enjoy, guys. Scireef will be on again. That's for certain sure. And let us know your feedback. Let us know what you think of Scireef. Let us know what you think of the corals. Drop us a like and subscribe. Drop us a comment. Interaction helps us every single time. Yeah. That'll do, won't it?
That'll do.
Cool. Talk to you soon.
Yeah.
Thank you so much for taking the time to come and join us today. it's a funny little industry here in Ireland, but there's a nice little core of reefers who are very passionate about what they do and very interested in the kind of furtherment of the hobby, the kind of the modern advances that are going on. And I know you guys are a lot to do with pushing the hobby in that kind of direction. So it's fascinating stuff that you guys do. So really interested to talk to you. And yeah, thank you so much for coming on.
So, can you tell us about Scireef?
Sure. So we should be able to do more.
You guys are polished podcast
Professionals. Brief like background, how everything started, where we come from. So we've been working with the corals in like a scientific context for the past 15 years. So back in the days, we used to fly to Guam, often to the University of Guam. So we started with our bachelors, and I mean, we don't have to go into that far back, but basically we travel to Guam almost every year for the mass spawning to work with the coral larvae and settlement and juvenile corals, because we had different projects in our like masters and PhDs and postdocs to investigate how different factors impact the embryo development, settlement, the juvenile corals, because these stages are so vastly different physiologically from the fully developed ones. And over the time, we got better and better to produce these larvae and settled recruits and juvenile corals. And then at some point in 2015, we had a tank with about 30, quite a bit more than 30,000 baby acros of about one, like a size of a cent, basically cent. And so everything for scientific studies, of course. And me being a lifelong aquarist and fascinated with corals, I thought and asked Marian, hey, you know what this could mean for the hobby, or what you could do with that if that would be possible to transfer it to the hobby. And at the time, Marian wasn't quite an aquarist yet, but a fully scientist, and she was like,
Not really. Yeah, Sam has been an aquarist like long before he became a scientist. So he had his first freshwater tank when he was like 5, 6 years old.
yeah, with the start of elementary school.
Yeah, so he was, and he had an reef tank also in his teens already. So he was a reefer before he became a scientist, and that helped us a lot in our scientific career. But I was really first the scientist.
Right.
Very good.
And that
You've gone reef keeping
I
to
learned
to reef keeping.
Yes. So it helped us a lot. And I really started to appreciate like how much more the reefing community knows about how to keep corals actually alive behind glass than scientists do. Yeah, and that helped us a lot in our scientific career, but also, yeah, then from the scientific career, we really thought, okay, we want to give back. We want to like renew this hobby side and bring this way of spawning corals and raising really corals from eggs in aquarium conditions and bring them into the aquarium.
Right, because that's something I think where that we probably need to track back a little bit. I know certainly from my kind of knowledge and experience of this, and definitely the guys listening, when we think of corals and coral propagation, most of our minds really just go to fragging, and that's about it. You take a large coral, you break it down into smaller parts, you put it onto frag discs, it grows into larger colonies, etc., and that's basically all there is. But that's obviously not the case. Corals come from eggs and fertilization in the wild, just as other organisms do. So it's a totally different thing, really. So I don't know what is how does a coral spawn then? What is that process?
Yeah, so there's really no way of reproduction that corals don't have, kind of. So, but about 80% of corals are broadcast spawning corals, and that means that most of these, once a year, they release eggs and sperm into the water column, where fertilization takes place in the water column, and they develop within a couple of days to little planula larvae. They're about, depends on the species, roughly a millimeter, and they have little cilia they can swim with, and they swim around, and then within a couple of days to weeks or even months, they can spend time in the water column, and then they settle on the seed floor and they undergo metamorphosis just like a just like a frog or a butterfly. From a completely different animal, the larvae, they become a coral with a mouth and tentacles and everything that belongs to a real teeming coral.
How different are they at that stage? Like, for example, obviously you'd be able to identify them, but so if you were to look at that larval stage under a microscope, can you tell at that stage that it's LPS, SPS, like whether it's a Duncan's or whether?
Well, when you know the species well, but it's hard. Like, I mean, it's really hard. Like even for us, I mean, we have worked with a bunch of different species now, and you can see like differences of individual, like even with tenuous, they might have a little bit different coloration, a bit more pinkish or orange. But then you come when completely different species, they might have the same coloration, but they might also be blue.
Yeah, it's really hard to tell, actually. Yeah, but generally the saddle ones are physiologically totally different. I mean, they're very sensitive to a lot of things because I always say they're tiny, they're helpless, and they're very tasty and nutritious. So there's a lot of critters out here that are looking for baby corals, but then on the other hand, they are still very plastic and adaptable, so they can be shaped by the environment, which kind of makes sense because if you look at how vast regions they could conquer over time. And if the larva is traveling like 3,000 kilometers to some new volcanic island, it's pretty unlikely that the conditions are the same as in the parental reef. So in order to colonize almost all Pacific, they
Life wants to survive, right? Life wants to adapt. Life wants to.
It's the cage for almost all juvenile organisms.
Yeah.
You look at like from a tomato plant, doesn't matter. They all undergo developmental phases that are shaped by the environment.
Yeah. And by the nature of the aquatic environment, this whole broadcast spawning is something that you kind of come across frequently, isn't it? In that kind of environment, you see these mass spawning events, and they coincide with mass feeding events, and whole entire animals are moving and relocating to various different parts around the globe to meet these spawning events, aren't they? Like, it's amazing, really.
Yes, and even the completely different species, they often do it at the same time because there's just that many fish that can like fill your stomach with all the eggs in the water, right? So you have like some worms that spawn at the same time as acroporas, for example. And I mean, it's a big feeding party for the fish, but there's just still so much biomass produced at that one night that once all stomachs are filled, rest, you can just move on to become a
I find it absolutely fascinating. I love watching documentaries, the whole Blue Planet, David Attenborough stuff and everything like that. But when you literally see, there's a million herrings in this part of the world at this particular time. Why? Well, because something else is spawning. It's amazing. It's amazing. So obviously this kind of coinciding of life and this whole kind of idea that these things happen at the right time means that they must be triggered by kind of external events and external things that kind of multiple different organisms and multiple different animals can react to. So from a totally layman's point of view, when it comes to spawning corals, that must be really difficult to recreate in a glass box, is it not? Because it's hard enough to recreate simple water that grows coral with light and flow, to actually get these animals to believe that it's their time to spawn. I mean, that must
basically they need to know what day it is. Because they all sit in the reef and can't move around, so they have to spawn at the same time. And there's several factors that allow them and their internal clocking mechanisms to determine what time of the year and what day it is. And that is basically a lunar cycle is like the base rhythm. So at full moon, you have the entire night. There is a light signal that they can
sense.
Yeah, sense, that's the word.
So how do you predict it? In a glass box, like,
So
can you set a time frame?
So we do the light cycle of the moon and also the sun, sunset and sunrise for a certain location, and then also the temperature fluctuation over the year. So that's kind of the seasonality. There's always even a little bit of a seasonality in temperature. And then for most species that we work with, not all of them, But for most species, we kind of have a rough idea when they usually spawn in nature. And that is helpful for us because that is kind of an estimate, okay, when they will spawn in our lab because we know what season it is here for them because we can just make it. And and then for many species, it's kind of easy because for our corporates, for example, we can break a little piece off and we can look at the egg development inside. So over a couple of months, the gametes develop in the skeleton or like in the tissue deep inside the polyp. And we can see really tiny eggs become bigger, big white eggs. And then shortly before spawning, usually they get, or like last 2 months, usually they get a little bit of a coloration or even intense coloration of like an orange or something. And that's when we know, okay, this coral will most likely spawn this month. And then it is a couple of days. That is usually where we are just like pretty much ready. Yeah, usually the 2 weeks after full moon where most species spawn. So we don't have like an exact day. We can't tell you, okay, this tenuous will spawn in November or on November 8th or something. That would be better. We're not quite there yet. Usually, because when we miss it, it's also really painful because we wait another full year for this individual to go. So they're pretty much always.
Right. Yeah.
And with the precise timing of the spawning, it's fairly easy to see because so it always happens after sunset, right? After full moon, so it's dark, and that is also an issue that it needs to be dark in that time between sunset and moonrise. So in that time frame, then we check with the red light because the corals, they can't really pick up the red light. And before they spawn, they get ready, and it's called setting. And the polyps inflate, and they push these little packets of eggs and sperm into the polyp without releasing it yet. And in the red light, it kind of looks like a Christmas tree with the balls on it.
Yeah. Yeah.
So these white dots in there, and then we know, okay, that colony is going to spawn within the next hour or so.
Yeah, some species are very nice and some individuals are very nice and they basically tell us like an hour and a half before they go and we can like have time to get ready.
Yeah. Amazing. Amazing.
Others are not that nice. They just go off and you're like, oh no.
Oh no, it's happening! Quick!
Can I do different species of coral have different behavior or spawning?
At different times, yes. Some are like Kentai and the tenuous complex is fairly early in the evening, which is very convenient. But others, say Humulus is fairly late. Other species are around midnight then, or even past that. So
Right. Yeah. So you gotta stay up.
Also multiple times a year actually. So we have some individuals that go several times a year or several in a couple of like 2 months in a row or something.
several days in a row.
Several days in a row. So it's always a little bit different.
Right. Yeah.
And you guys are doing this to produce corals for the aquarium trade, basically, for people can buy corals from you, right?
Yes,
So
because we really
have them shipped.
Yeah. So the first ones we started selling in November of last year, and we started friends coming from the university context. And doing it on Guam is a whole different story than doing it in the aquarium, right? So in Guam you get the water from the reef, you got the moon from above, and the sun is also free. So that's really not too complicated. Then of course all the processes after are really complex and complicated. But then in end of 2019, early 2020 was this pandemic creeping around the globe, and there's travel was open. But luckily at the same time, so we said, okay, the research must go on. At the same time, Jamie Craig's from Horniman, who was the first one actually to spawn coral in captivity, he started a spin-off company that is putting together systems that can allow you to do the climate simulation. And at the time, we started a collaboration project with Tropic Marine, and they also supported our research for a long time before that. And together with them, we then were able to obtain systems and had them built into a shipping container that we then put up at the university.
Wow.
And in that container, then in 2020, we had the first spawning, first successful spawning. And from then on was a couple of years. We always was still research context to optimize methods for like upscaling. And then in 24, we got a funding for like a transitioning project to go from scientific research into commercial application. And that's also when we launched the Scireef. And yeah, ever since then, built up the facilities here and started to produce corals for the trade.
So
Yeah.
what's
So
the...
especially
Sorry, go on.
you go on.
No. I was going to ask a different question, so I'll let you go first.
Okay. Yeah, so during our scientific work, we really got to know the juvenile corals much better, and we found so many ways that they are more able to adapt to their environment. We kind of tapped into it a little bit, but we really thought, okay, that would be so useful for the aquarium trade, because right now we basically take wild corals, and some are better adapted to our aquarium conditions, and we can hold them relatively well. Some are difficult and very challenging, and some we're really not able to keep well in aquariums at all, and we don't really know why. But there is definitely a lot of challenges, and and especially like acroporas, for example, that we work with a lot. They're still very challenging for many reefers, and there's a lot of like can be very discouraging when you when they grow well for a little while and then not so much anymore.
Right.
And there is like a huge benefit when they grow up under aquarium conditions, and we can shape them already a little bit in that environment, that they're just better able to grow under regular aquarium conditions.
That's an effect that we've been seeing in fish. No, I mean, now you can buy mandarin fish that you can feed with like dry food. But like 15 years ago, nobody would have believed that.
Yeah. Yeah, it was unheard of.
It's good. That's actually on the same train of thought. I was kind of going in that direction, which is, everyone knows the advantage of tank bred fish. A fish that has lived all of its life and all of its development in that environment is going to be more adaptable to it. I have a beautiful tank bred Centropiga Rentio in a quarantine at the moment, going into a tank, which is just the fact that they're tank breeding that kind of fish means that a fish that was previously very difficult to keep is now a lot more adaptable and a lot more easier to get into a system. And that's the real advantage, is it? It's the adaptability of the coral that people get. That was going to be my question. Why?
That is a big
It's just fresh, personally,
Why not? Yeah.
Yeah.
And then second part is the genetic diversity. Especially in species that tend to have a wide coloration variety, like tenuous and kentai. So we see that in the offspring, that there's so many different varieties, and you can't even predict from the parents' coloration what is going to be in the offspring. That's always a black box. But it's really amazing because if you think about it, like every fertilized egg is its own genetically unique individual, right? And they're all a little bit different,
Right.
and that's always fascinating to see what comes out. And you can't see in the beginning, because when they start to take up the zooxanthellae, they're all still very tiny and brown,
Right.
and you have no idea how they're going to look like. But then after like 3-4 months is when the first pigmentation comes, and before that, and we had and look at the setter recruits with zooxanthellae were super hyped about it, and they look in the tank like,
Looks like Arcasia.
Okay.
Right.
Like
It's almost like I want what I'm thought
like I was gonna have.
Very good.
Yeah. From what I've seen, some of the corals that you do produce, they how do I say, they become hybrids. They have like 2 species of or
Mixed together.
Yeah.
Can you tell us how
Yeah,
that
so
happens?
So there's 2 different things. So hybrids are juveniles of 2 different species. We try to work on that too, but it's really hard. For example, if you have like a millipora and a I don't know, humulus or something, and to produce eggs, fertile, larvae from it is really tough. So we kind of work on it, but we haven't gotten very far. Most of the time, like where we get usually like 90% fertilization rate, we get maybe like 3% fertilization rate, and from those 20 fertilized eggs, we get like 3 larvae and just 1 settles, and then that one dies.
Or something
So
that we had recently with the Moricata parallels, right? You get nice fertilization and they develop and then they turn into larvae and then
like everything stops.
they don't develop further. And
So that is hybrid. But what we can do quite a bit is chimeras. So chimera are 2 individuals that become 1 individual. That happens when 2 larvae, usually of the same species, they settle right next to each other and they fuse to become 1 animal. But they have both their distinct colorations. So especially, for example, in like tenuous or kentai, you have like orange, green, yellow, whatnot, and blue and turquoise or different colors. So the 2 different size can be completely different, and when they grow together, you get these beautiful spliced branches where the colors just start to swirl, and we get that sometimes from in the wild as well. This is nothing that can only happen in the lab. We sometimes very rarely find a chimera animal in the wild gets introduced in the aquarium trade, and we have a couple of that already passing around. But in a lab environment, you can force this much better.
Right.
So I would say we probably have the majority of chimeric individuals of the world in our rooms here. So that it is really exciting, and it's we're still at the beginning of playing around with this more. So it is a bit difficult to tell these little guys to settle next to each other, 2 or 3 only, because when a lot more settle next to each other, they die off like always. You never get like an animal, or we never gotten an animal made from like 10 larvae or something. So 3 is probably the max we ever saw surviving. So we want like 2 or 3 larvae settle next to each other, but not more. So we have to play around with it a little bit. But it's so exciting, like to see these individuals, because that is so rare right now, and which it is also a little bit rare here. And there are still the few we have, or with the few we are selling at the moment, are quite pricey. But we will try to get better at it.
And to look back to the actual hybrids, or no, you go first.
No, go on. You can, and you want to ask a different question. Go on.
One species with the Fijian tenuous parents, we had gotten them as tenuous. They look like tenuous and spawned, and everything developed nicely. And then in the offspring, we had a huge variety of morphologies. Like some have these really long corallites that look more echinata-like. And we have found that like mixed form, and some look more like actual tenuous, and that within one batch. So the thought, was still waiting for genetic data. The thought is that the parents we got, it looked like tenuous were hybrids between tenuous and echinata, because even though they're looking totally different, they're within the same
Genetically, they're quite close.
within the species complex, if you want to say, or you know, if these were or had some echinata traits in them, and then they are crossed again, then in the next generation you get like an uneven distribution of the traits. So some get a lot more of the tenuous genetics, so some are intermediate, and some get more of the echinata. Or that's, I have no idea. Hypothesis.
Because there's a lot of that in reptiles.
And I mean,
You know, and they can, one trait can express more strongly than the other, depending on the kind of genetic code of the individual. Right?
yeah, and it's super exciting because from these guys, we actually have some that look extremely close to Echinata icefire, but we call them tenuous icefire now, and they grow super well in their career. And it's like the one thing, like everyone wants these Echinata icefire, but they like yeah, show me someone who like keeps them alive and growing.
I've put a lot of thought.
on fathom. It's really hard. Like you even like when you look for it, like to show me a picture of a frag that has grown over the frag plug. It's really hard to find, right? So, but yeah, we have them now from their tenuous parents, which is very exciting.
I hopeful that at some point we'll get the genetic data. So our colleagues at the university took samples a while ago, so it takes a bit. So, but at some point we'll hopefully know for sure what it is.
Very cool. Yeah, very cool. So, what I'd like to know is how when these spawn on events happen, how do you actually collect the larvae? How do you collect them from where? And how, do you put them into a new system? And what is the process?
It's a long process, and
Yeah, I'm sure it is. I'm sure it is.
The abridged version of getting a larva into a frag plug. Really brief, really brief. Well,
so the spawning time of the year often coincides with the phase when the water temperature is rising. Right, so in Australia, it's usually starts in October, November, December are the main months. Fiji is a little bit earlier than places like Saudi Arabia, the Red Sea. They spawn in spring when the temperatures are going up. So we know kind of the season, and we can also check when the eggs are ripe. So, but once they got ready to spawn, then yeah, it's showtime. And we, of course, then turn off the flow pumps. And to separate, to know which eggs come from which colony, we place like the acrylic pipes over the colony that goes above the surface. So the egg bundles, they contain so much fat that they are really buoyant, so it goes up and stays within that acrylic, and then we can collect the bundles from there, right? And we do that from the different colonies. Hopefully it's more than one that spawns. And then after about like an hour, depending on temperature and species, that lipid film that keeps that bundle together dissolves. So we collect them, have them in cups or bowls or whatnot, and then the membrane dissolves, and the eggs are staying at the surface, and the sperm are in the water. And then we mechanically basically separate the eggs and the sperm, and then do fertilizations of like sperm from colony A and eggs from B, right, in the right concentration, and vice versa. And then usually some species are quicker than continuous. Like after 1.5 hours, you can see that the round eggs, they start to look like a peanut, and cell division starts, and then you have the 2-cell stage. And once you have that going, and hopefully all of the eggs are now okay, as well. And then we transfer them into certain floating containers, like basically
Usually in a different system than the parents were, so we basically keep everything from that stage on already separate from the wild caught parents, basically, or mostly.
yeah. So the containers are basically like a larval chrysler, but horizontally.
So it's just like a bowl with a little bit of a mesh at the bottom.
Yeah, the eggs or the eggs are on the surface, and then there's a light water circulation that they keep moving, right? And then yeah, within about up to a week, they develop into larvae. So of course you have to take care that like embryos that die off need to be removed, otherwise you get like the fat and all that. And after a week, then they start to, because you're in development, they use up all the fat, and then they lose buoyancy, and they have their little hair and can cruise around, and then at some point they reach the bottom.
Right.
And when they're like actually exploring the substrate, then you know, okay, now it's time for them to settle. Yeah, that process we also been researching for.
Yeah, that was quite a lot of our focus in our scientific life. Like what makes coastal? We know it's the bacteria, like the bacteria that live on choline algae that induce the settlement process. And we kind of isolated the individual bacteria, found out, okay, what species of bacteria is it? And then from that bacteria, we isolated the chemical compound that actually induces the settlement. And that was kind of a long work, and it was not just the two of us. It was a whole working group that worked on it over like a decade to find that out. And But that gives us a huge opportunity because by now most scientists and people that do coral spawning, they use crystals coralline algae or like a natural biofilm on some kind of substrate where they settle, and it works sometimes better, sometimes worse. You never really know like how well your biofilm of the day will work. That is an issue. And whenever you have a natural biofilm, and especially crystals coralline algae, they don't like to be settled on. They want to keep that space for themselves. So they have defense mechanisms, and there's all kinds of little organisms that like to snack on these little tiny defenseless corals. So survival of juvenile corals on natural biofilm is usually really low. And that is, of course, a huge problem when you want to make a business out of it. So we can use this extracted compound to have the coral larvae settle on completely clean surfaces. And that gives them a huge advantage because there is no neighbors that fight them and eat with them. So they can. We have them then in a normal system with like a whole biology in the system, and that of course this clean surface gets settled on by bacteria and algae eventually. But for the first weeks and months, they have a huge advantage. They can grow, they can take up symbionts, they can they just survive and grow fast. And this faster growth at the beginning, that gives them so much more strength and they become bigger much faster. And that is a huge advantage because if we wouldn't do that, we would probably have to wait for another year to get them to the same size as we get them now within like 9 months.
How fast do they grow? How fast from larval stage settling onto wherever you want them to go? It's amazing, I think, by the way, that you literally identify a bacterial strain that you can then provide them to say, I'd like you to settle there, please. And they go, yeah, okay, well, that's a great environment to settle on, so we'll do it. That's wonderful. Well, I was thinking in my head, how do you get this from larval stage to frag plug and then from there to growth? So how long is that process? Months, years?
Depends very much on the species. So,
Sure.
like again, the tenuous Kenti, they are relatively fast. So they are really nice because they, even when they're really, really small, when they're just like 4 or 5 millimeters, they already start to grow three-dimensionally. They grow up and they make these really like tiny little cute colony shapes, and they also get their coloration really early on. So about like 9 months in, they are already like this size, and we started selling after a year and they were already like this size. And by now, like they are a year and a half now and they are like this.
Wow.
So
Full colonies in the modern trade.
yeah, they are like full good decent sized colony by now. So yeah, last year, imagine like they grow so much faster than what group is used to because they're juvenile animals. Juveniles grow faster than adults. That is just for any animal that is true, and also for corals. So yeah, imagine like they were a year ago, they were just like 4 or 5 millimeters, and now they are this. So they double their polyps every like 4 to 6 or 4 to 8 weeks, I would say.
Wow.
Yeah, we had to actually slow them down in between because we settle them on like plastic plates and grow them out for a bit, and then we transfer them to our own plugs. Right, we have the PVC plugs that are also they get their NFC tag in there. And these things that took a little bit longer to produce than we hoped for. So it was an end of August until we finally got them, and
it got really crowded on the baby plates. They were still on, and we're like, oh no.
and at that age, then they start to fight when they touch each other. So I turned down the light a little bit and less nutrients, less feeding, so they don't grow too fast.
Yeah,
Right.
but other species, they take longer. Like the millipora, they grow maybe half as fast, and also the coloration doesn't come in for a while. Like it takes like the year. They're basically brown for a long time, then they all become like super green.
Yeah.
Yeah, well, yeah, okay. Or also the spatulata. Like we have like all kinds of different color parents, and the juveniles right now, they're just green. like pretty green, but all green. But I'm sure like half a year from now, they will look very different.
Yeah, Millipora always strive to, so a Teniochis starts to do the tiny colonies early on. Millipora goes flat, like
Right.
pretty much
Encrusting as long as they can before they start branching.
it turn brown, and then the branches come up, and then the colorations come.
That's amazing.
So, one thing I was interested in, and just thinking about it, when these aquaporta actually do these larvae settle and they pick up a bit of place in this plug and stuff like that, how do they eat? Like, and can they produce like enough Zeus and Zelly to kind of sustain them?
That was another thing we had to work with. So at first, we just use basically the symbionts that are in every aquarium, because every healthy coral produces more symbionts than they need for themselves. So they just put some out. But our filtration system that we need to keep a healthy tank environment is pretty good, and it takes out many of these symbionts. And turns out like it's not enough, or we found out it is not enough for the juvenile. So they don't have any symbionts at the beginning.
Okay, yes.
Or we work with, so zero. They have to take them up from the environment and introduce them into their tissue. And yeah, so we started to grow these symbiotic algae in flasks. So we have different symbiont cultures, and that is actually quite tricky to keep them in culture. Some grow really well, but they are usually not the kinds that Aquapora like as symbionts. And those kinds that they like, they are really hard to keep in the flask, of course. So it was quite a challenge to get a good variety of symbiotic algae that we can then introduce and offer these juvenile corals early on. But it's also a huge advantage because, for example, the temperature range the corals are adapted to is very highly influenced by the type of symbiotic algae they keep. When they grow up on high temperatures, they have certain symbiotic algae in their tissue, and when they're adults, they can't change that anymore. Even when they come in an environment where it's colder and other symbionts are available in the water column, they can't just pick them up, put them in their tissue. Like once this symbiosis is developed, they just recognize this one or two max symbiotic partners as symbionts. Any other symbionts they pick up, they just digest them.
Right.
So yeah, when we get corals from really hot environments and put them under 24, 25 degrees Celsius, it is a little bit stressful for them. So we offer them symbionts where we know they work really well under like our typical water parameters, the typical temperatures that we have, and we give.
And is that regional as well? Is that regional around the globe as well as to where these where the corals might come from and where the symbionts might come from? Are you matching kind of in a way?
Yeah, it says in the symbionts that are present in the oceans, like in the Red Sea, a bit different from the Pacific, but there's a huge variety of them. There's, I think, at the moment, like 120 different ones are described. And no, we know any more. There are, most of them you can't really color. Yeah, so there are differences. Yeah.
But of course, our goal is to make healthy corals that grow well under aquarium conditions. So we will never like grow corals here that we will bring out in the reef. So we're not really concerned if do they get the right symbiote from that region.
Yes.
We just want one.
Just want to know which one it needs. Yeah.
Also, we just offered them a bunch of different ones that we think should work well, and then they can use whatever they feel is best.
Great. Thank you.
Well. And so I've recently seen that you don't only spawn aquapora, you've started spawning other types of corals as well. So can you tell us a bit about that?
Yes, of course. So, of course, the aquaporas, you can just snip off branches, and that works well, and that will always be the case. And everybody should do that when you have a really good adapted coral for the aquarium. But of course, with some corals, you can't do that. And we had never worked with other, or we had worked with some, we have had worked spawned some other species or genera, but not the large polyp ones that you can't branch back. So but we started to work with some, and we were pretty successful now with Trachyphilia actually. So a couple of months ago, we had a big Trachyphilia spawning, and everything was really different with the Acroporas. And of course, we didn't think we could make it work first time around, because like for the Acroporas, we needed like a really long time to get good at this. And there's still a lot of improvement that we need to go through for the Trachyphilia as well. But right now, we are pretty happy with like the amount of juvenile Trachyphilia we have and how fast they grow.
Yeah.
Like it was really interesting. Like when I saw the eggs, I didn't even want to continue to work with them because they were tiny. They were like a fraction of a they were a fraction of a millimeter. And Sam was so excited. And I looked, I was like, we can never make this work. They will be like a tiny egg means a tiny larvae means a tiny, teeny tiny polyp at the beginning. And the Acroporas are already super senseless and helpless. And they need a long time to come, like about 3 months till they're like able to deal with the world. And I thought, okay, the Trachyphilia, that is horrible when they're so tiny. But it turns out we had good instincts, and a lot of them survived. And we had to figure out this settlement, for example, completely new. The compound that we used, we couldn't make it work on, but we found other ways to settle them on clean surfaces pretty well.
Yeah.
Yeah, and they survived. Like a lot of them survived, and they grow surprisingly fast.
Yeah, it's really amazing to see how quickly, like after each feeding, in the next couple of days, you see a growth spurt, basically. So I don't know how long that is gonna hold. A different, either like live zooplankton, like or also brine shrimp, then a blended mix of basically everything that lives in the ocean and run it through the blender, and then they can pick what they need.
It's eating fruits there as opposed to not.
So some what's that? There are phytoplankton as well. So a variety of different things.
What's the uptake of nitrate and phosphate from the water column like in young corals or any corals from your guys' research?
They draw quite a lot. So the big, so we have 7 systems of like 2,000 liter here, and 2 smaller systems. And of these 7, 4 are for rearing the juveniles. 3 are the parental tanks, where we do all the climate simulation, and that is also not wellness for them. So in the summer month, it gets pretty hot, and you see the consumption of everything goes down. And then Australian winter, it's pretty cold, and then also consumption goes down again. But in the broad system, we run usually at like 25 Celsius constantly. And the system that is the densest stock that draws more than 3 degree DKH a day, and like 0.15 milligrams of phosphate every day. So that's what we have to replace. Okay, and then nitrogen we also dose in different forms. So either like the tropic marine products, but also like ammonium chloride and urea. And we use nitrate as an indicator of how much we can put in. So if nitrate goes up, then we reduce the input. If nitrate goes down, then we can put in more.
Okay, but you're introducing nitrogen compounds in the form of urea, ammonia, rather than in the form of nitrate, right?
Yeah. Because that is easier for them to utilize.
Yeah, corals are
And,
pretty bad at eating nitrate, right?
Yeah. I mean, it works, but it's more energy intensive.
Right.
Yeah. So we use been using that for quite a long time. Yeah, and phosphate is always something that we keep an eye on, that we don't go too low.
Yeah.
We keep phosphate at a target is around 0.1 milligrams. So if it goes down to like 0.05, then we're getting nervous.
Not that that is a bad parameter. If you run a system like that, that is completely fine. Just for us, because we have so much consumption that when we go below that, then there's basically one of the tanks, there are corals in there that have zero, basically. So chances are really, really high, and we don't like to go into like a zero phosphate for any of our corals. That's like definitely over the many years we kept acroporas. We figured they don't really like that. They don't go well. The immune system becomes worse. They start to get diseases. So that is definitely something we rather keep on the higher side.
So, and are you pulse feeding with your nitrogen compounds, or are you trying to maintain constant level?
Yeah, once a day.
Yeah, that goes in once a day, so we get like a one peak in the morning.
Not that we think that is best. It's just more.
No, I actually think it's good than spreading it out into little small doses, because I cannot say which organism is or how good they are at absorbing it. So maybe some are faster. So if I put in just a little bit, the idea is
You're just
and
get taken up by bacteria.
all the microbes very efficient at taking that up. So by giving like a bigger peak, I'm kind of sure that all of them have the chance to obtain some.
I always think about kind of how the natural situation would be on the reef in terms of pulse feeding and feeding of corals of particulates and particulate nitrogen and phosphorus compounds and stuff like that, rather than this constant level of nitrate and phosphate. And the words of Luakis ring in my ears about how they're not exposed to a constant level of these things. What happens is a massive great shoal of parrotfish or chromis or tangs swims over the top of them, and they all go to the bathroom, and the all of that rains down over the corals, and the next wave hits and away it goes, and they're back to clean fresh water. So I know there's a lot of talk about pulse feeding of particulates.
I forgot which, but there is a study that looked at exactly that, and there's often there's like two peaks when the fish activity then picks up in the morning. Then there's one peak, and then towards the afternoon is another peak. But
Well, I suppose you're looking for corals that are adaptable to things that we can do, right, rather than copying what we can't necessarily.
Yeah, that is another thing. Like, on one hand, it's also more feasible for us to do some things per hand than rather than adjusting pumps every day. And we also don't want them to always constantly have the absolute perfect conditions with zero fluctuation, because that is not what 95.
That's not the hobby.
Yeah, it's not the
Exactly, that is not the hobby. So we want our corals to be tough and also to deal with KH fluctuations and fluctuations in all kinds of water parameters. Even though we try to keep them somewhat stable, but we don't like have for example, like an automatic dosing system to keep alkalinity exactly at the same level. Like we have dosing pumps, but then we measure per hand every day.
yeah, so don't
There is
trust
fluctuation.
the automated ones.
Right.
Yeah, you
Yeah, you wouldn't want to risk your decade of work and research on a machine.
Yeah, broken probe that then decides to pump in like 5 liters of alkaline.
That could be a problem.
could be
That could be a
devastating.
problem. Do you know what I really love? I love the thing that you guys do with the fact that you can scan your phone against any individual coral and you can pull up its essentially birth certificate, information, where it was born. You really are producing individuals with information attached to each particular piece rather than just a chip off someone else's. Where does that idea come from? That's an amazing brainwave to put that information in digitally onto the coral.
Multiple aspects.
Yeah, first it was just like, how do we keep track? When we have the same species spawning several years in a row, how do we keep track of like which coral is from which spawning, how old is this individual? So we thought, okay, we need some kind of system to keep track of this. So NFC chips was right away a good idea that we had. And then we thought, okay, we could make little plugs where we can push the little NFC chip in.
Yeah.
And then every customer also has this kind of information. So I mean, we are not like with birds or something like you have to have a ring on the feet that is mandatory. With corals, we don't have to like track each individual. but it is possible nowadays. And yeah, I think it's a cool feature to have, and it's actually not that hard to do with these NFC chips. They are super cheap by now. So
I mean, the way Sigyn did it, it escalated a bit from the original thought.
yeah,
So, yeah, we have we have an own app that at some point will also be publicly available, where you can basically do a little family album of your corals and put in pictures or whatnot. But generally, so when we scan and write on the chip, then it corresponds with our invoicing software and all that. So that got more complex than Bill. Yeah, very responsive.
Tell us a little bit about the Tropic Marin involvement. Then you guys work with them. They help you. Go for it.
Yeah. So when we had the idea of basically bringing this kind of coral breeding to the aquarium market, it was clear that we can't do it ourselves because that involves a huge investment, and it's just too big for the two of us to do by ourselves. So we started to write a lot of like articles and like Coral Magazine and go to
Fear exposed
expositions, conventions, where we kind of introduced the ideas, and that's when Tropic Marin came to us and said, okay, this is amazing. We think this is really the future of reefing, and they, as a big player in the market, they saw also a kind of responsibility to make the market more sustainable and future-proof, because I mean, at the moment, there's really corals come from Indonesia and from Australia, and there's a very little, like a little bit from Fiji, but that's it. And there's just very few political decisions that need to be made to shut that all down. And we would not get any more. I mean, we see it already. A lot of species are not available. And that can easily, like one big bleaching event in that region, and maybe the politicians there decide we stop this industry now, and we would have a big problem. So that's what really they thought this could really be great if we could breed corals here in Europe for the aquarium trade and be independent of wild collected corals. And they started, it was a long way for them and for us. So they first started to just support our scientific work to get all this knowledge about how to settle corals and how to make many of them, the juvenile corals, survive this.
Yeah, and even before that, they just started to support the research with pulp, basically. And just back until 2016 at the university, we were still using water from the North Sea.
Yeah.
Right.
Poured artificial salts.
As we we treat it. You can keep corals in it. So we then remove all sediment and then run it through ozone and UVC and big skimmers and then send off an ICP. And then sometimes you have like 100 micrograms of zinc, and then we dump everything again and pump again. Right. And then sometimes the ICP looks great and you use it, and then the corals are just about something.
And you don't know what it is, probably some organic compounds in there that we can't track.
Yeah, so
we were
the compounds in salt water that you're not able to trace is having an effect on your.
If you measure it, then you get like 600 peaks of stuff in there, and then we still don't know what's going on.
Yeah, so we couldn't successfully keep acoperas in the North Sea water for a long time. And so they started to supporting our research in 2017. And then they got even more involved with acquiring this container where we were able to spawn corals then. And they are just now, they're amazing in supporting our work. They're also like really our business mentors, because we're both scientists. We have like, this is business, so this is new for us. So there's a lot of work in that side we have to do. And they're always just a phone call away with some of our beginner questions of like how to successfly run a business. And
there comes a letter and I'm like, what does that word mean?
Yeah.
I kind of ask them so.
Yeah, so we wouldn't be here without Tropic Marin, and we're immensely grateful. And they are a bit quiet, I think, about like how much impact on in the back of the house in industry, but it's huge.
Sure,
Yeah,
great company. I love the product.
Yeah, they have really good products, and we're pretty much exclusively using their products. We have a couple of things we mix ourselves, but besides that, we're using something else.
And you're using the same stuff that's available on the shelves. You're spawning corals inside the lab settings, and you're using the stuff that people buy in. And
We get it in the buckets.
Yes.
Yeah, sure. I'm sure you do.
Since 2016, and yeah, that one we usually get in either 20 or 30 kilo bags, but it's the same stuff. The Pro Reef.
yeah, yeah, brilliant. Yeah.
The Tropic Marine Pro Reef, and then also we use the standard 3-compartment belling system from Tropic Marine and the AEP elements for the micronutrients. Yeah.
Another ICP and the block system.
Yeah.
As well. So
Yeah, and that is also really cool because when we have some ideas for products, it's also really fun because like you have been really involved, for example, in the block system for like the ICP components to develop that, and that is also
involved in like fair research development for quite a while now. So that we can bounce back, work ideas, and then came up with the stuff. So no, it has been a absolutely fantastic cooperation.
Wonderful, wonderful. Where can people find more about you guys? Where can people find your work? How can people get your corals? All of that kind of stuff.
So our website is still work in progress. You don't really have to look on the website. That is still very, very basic. But we do give like daily updates. We're really active on Instagram. So if you follow us on Instagram, there's really all the information out there. We do YouTube videos quite regularly, also on the Tropic Marine channel. If you, there's every week there is a new video on the Tropic Marine channel, and very often it is about us and our work. So that's, and we're also on TikTok and I share some videos on TikTok. But Instagram is really where you get everything. And that's also where we share where you can get our corals at the moment. So we usually don't sell corals to
B2B, basically. So for logistics, and that would be too much. For and this says
we almost exclusively do B2B. So we have a bunch already, about 10 coral shops that sell our corals in Germany. We have one in Austria now, and you said we might want to bring some.
It almost sounds like you guys could use over in the side of Europe as well, doesn't it?
We don't have any of our corals in Ireland yet.
Yeah, we've been definitely doing this, and I've been talking to some customers, and I've been saying to them that we were planning to speak to you and stuff like that. There's one or two guys that are really excited, so they think they would like some of these corals too. So
it's the future. It's the future. Everyone has to get on the back of this stuff. Like you say, the rock could be pulled out from under us at any point. So what you guys are doing is what's going to keep this hobby alive in decades to come.
Yeah.
Thank you. Yeah.
Fascinating, wonderful, guys. Thank you so much.
Yeah, it's been amazing, really, and we'd love to have you on again in the future. Maybe.
Yeah, we'll be fun to talk to you.
Maybe when these Wikipedia as well come in to get a little bit bigger. Maybe we can get our hands on some of it.
we hope for like any towards fall or end of the year that they're big enough to go out in the.
Of course, they won't be as big as people are used to from like wild collected ones, but they will be a decent size.
Very cool. Have you any other corals coming up or anything else that's in the works?
So we have quite a few Acrophora, different species in the pipeline. And then also regarding the chimeras, we have quite a few things going on. We have more LPS, but that is, in the very early stages, let's put it like that. So we have Canthophylla, parent colonies, Scoleus, Cinerina.
Yeah, we will definitely try to work much more with the LPS and not only aquapods, but we will still continue to like breed more beautiful Acrophora still.
Amazing stuff. Amazing stuff. Okay, guys. Thank you so much indeed. Thank you so much for coming. It's been an absolute pleasure, and look forward to doing more with you.
And thanks for all your time and this chat. Really, take care, guys. All the best.
Thank you so much for having us.
Thank you so much. Happy reefing. Keep it. Take care. Bye.
Bye.